Hello everyone, and welcome to this lesson, an introduction to DNA and protein synthesis. As we know, living organisms tend to always be growing and changing, and so they need to replace damaged cells and other materials within their body with healthy ones. In order to do that, organisms need to have the ability to synthesize new DNA and new proteins for the new cells and materials that it's using to replace the old damaged ones. So, as you can see, both DNA synthesis and protein synthesis are essential processes of life, and without them, there could be no life. If an organism wasn't able to do these things, then it wouldn't survive, because the new cells that it uses to replace old ones need to have their own copies of DNA. So DNA synthesis is essential, and old degenerated protein needs to be replaced with new protein. So without these processes, life couldn't exist. Now, both of these processes happen during a part of the cell cycle called interphase. Just to remind you, the cell cycle is divided into three main phases: interphase, the M phase, where nuclear division occurs, and cytokinesis, where the cell division occurs. Now, DNA synthesis occurs specifically during the S phase of interphase. The process is called semi-conservative replication, and we'll talk about what this means when we discuss DNA replication. Protein synthesis mainly occurs during the G1 and G2 phases of interphase. These are both growth phases for the cell, so you can remember G for growth. And it's during these growth phases that the cell is actively going to be synthesizing proteins. Now, when we talk about DNA and RNA, these are what we call nucleic acids. The Na in DNA and RNA stands for nucleic acid. So for example, DNA's full form is deoxyribonucleic acid. These nucleic acids are themselves comprised of one or more polynucleotide chains. As the name suggests, a polynucleotide is going to be a chain of many nucleotides. Poly means many. So if you have many smaller molecules which are called nucleotides together in a chain, that is a polynucleotide. Now, if you look at the individual nucleotides, this diagram here shows you what their structure looks like. To construct a nucleotide, the first thing we need is a pentose sugar. A pentose sugar is a sugar that has 5 carbon atoms. Think pentose like pentagon. A pentagon has 5 sides. A pentose has 5 carbon atoms. Now, if you attach a nitrogenous base to this pentose sugar, then what you get is a nucleoside. Then, if you attach a phosphate group to the nucleoside, this is what is called a nucleotide. So a pentose plus a base is a nucleoside, and a nucleoside plus a phosphate group is a nucleotide. Now, these nitrogenous bases on the nucleotides can be divided into the pyrimidines and the purines. The purines are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. One memory device that you can use to help you remember this is that the word pyrimidine has a Y in it, and the two pyrimidines, cytosine and thymine, also have a Y in the name. So you can remember, if there's a Y in the name, it's a pyrimidine. Now obviously this isn't perfect because uracil doesn't have a Y in it, but it can still help you somewhat. When two nucleotide chains come near each other, these nitrogenous bases tend to form hydrogen bonds with each other through a process called complementary base pairing. With this, what you have is that purines always pair up with pyrimidines. So you won't see a purine with another purine. It's always going to be pairing between purines and pyrimidines. In DNA, you have adenine pairing up with thymine and guanine pairing up with cytosine. In RNA, adenine pairs up with uracil, while guanine still pairs up with cytosine. When adenine pairs up with thymine or uracil, it forms 2 hydrogen bonds, and when guanine pairs up with cytosine, it forms 3 hydrogen bonds. This was just some background information on nucleotides and polynucleotides. So, nucleic acids are made of one or more polynucleotide chains, and polynucleotides are made of many nucleotides. Now, not too different is the structure of proteins. Proteins are made of one or more polypeptide chains. Polypeptides are themselves a chain of many amino acids. Now, let's come back to DNA for a minute. The DNA is what contains the information that our body uses to make polypeptides. The particular sequence of nucleotides in the DNA molecule that is going to be used to produce a polypeptide is called a gene. And typically, one gene codes for one polypeptide. So within a specific gene, you will find the information that's needed to produce a polypeptide. And that information is present in the form of the sequence of nucleotides. Based on the sequence of nucleotides, our body will put together amino acids in a particular sequence. It's the sequence of amino acids that's going to determine the properties of the final protein that this makes. And since organisms are mostly just made of protein, structurally speaking, the information in the DNA basically controls the entire organism. For example, proteins are important for the structure of our skin, our eyes, and our hair, and it's proteins that play the role of enzymes in our body. Now, let's look at a big picture view of what's going on. So, a particular sequence of nucleotides that will code for a particular polypeptide is called a gene. So you have a sequence of nucleotides that come together to make a gene. And you have many genes that will come together to make up the molecule of DNA. So each of these little colored bands here represents a gene, and each gene contains a specific sequence of nucleotides that will code for a specific polypeptide. When our body wants to make a protein, it's going to use this DNA molecule as a template to make a strand of RNA. This process is called transcription. So you can think of it like the cell will transcribe the information that was in the DNA into another form, RNA. Specifically, the type of RNA that we're talking about here is messenger RNA, or mRNA for short. From here, our cell will then use this mRNA as a template to put together amino acids in a particular sequence to form the polypeptide. This is called translation. You can think of it like the cell is translating this information from the language of nucleotides to the language of amino acids. This concept, which is that the flow of genetic information goes from DNA to mRNA to proteins, is called the central dogma. The DNA will first be transcribed into messenger RNA, and then the messenger RNA will be translated to make proteins. It's the proteins, which will be made using these polypeptides that you get from mRNA translation, which play a number of structural and functional roles in the organism. Since the properties of the protein are determined by the sequence of amino acids, and the sequence of amino acids is determined by the sequence of nucleotides in the DNA, ultimately, it's the DNA that encodes all of this information about the organism. This is why the nucleus of the cell is called the control center of the cell, since that's where the chromosomes are, and the chromosomes have the DNA. Now, once the DNA has been transcribed into mRNA, and the mRNA is then translated, what you end up with is a chain of amino acids. This sequence of amino acids is called the primary structure of the protein. Once we have this, the protein still has to undergo various levels of folding to achieve secondary, tertiary, and in some cases even quaternary structures. Okay, so the primary structure is just the sequence of the amino acids in this polypeptide chain. The secondary structure is going to be the initial folding that takes place because of hydrogen bonding, for the most part, between the amino group and the carboxyl groups of the amino acids. So, if you remember, amino acids have an amino group and they have a carboxylic acid group. And these different groups will interact with each other between the different amino acids in the chain to give rise to this initial folded structure, which we call the secondary structure. The most common sort of shapes that arise out of this initial folding for the secondary structure are called alpha helices and beta pleated sheets. For now, just remember these names. Once we have the secondary structure, the protein will then further fold to form a tertiary structure. This is going to be a result of the interaction between the different R groups of the amino acids. Just to remind you, amino acids have amino groups, carboxyl groups, the two of which we've talked about for secondary structure, but they also have side chains called R groups. It's the interaction between these different R groups in the different amino acids that gives rise to the further folding that we see with tertiary structure. This gives the protein a nice 3D structure at this point. Now, with some proteins, it doesn't even end here. Next, you can have a quaternary structure. This happens when you have more than one polypeptide interacting with each other and sort of binding to form a quaternary structure. Or it can happen when a polypeptide binds with a non-protein component. Basically, if you are adding something else to the single polypeptide, then you're giving it a quaternary structure. So whether that's multiple polypeptides binding with each other or a non-protein component binding to the polypeptide, either way, that is the quaternary structure. Okay, so we get how that works. Once we have our sequence of amino acids, we understand how it progresses to form a secondary, tertiary, and quaternary structure. But let's back up for a second. How do we get that sequence of amino acids in the first place? Yes, we mentioned already that we get it from DNA, but how does the sequence of nucleotides in the DNA end up translating to a sequence of amino acids? To understand this, we first need to go through a few terms. So I'm going to list them out first, and then we'll go through them and see what they mean. So you have the triplet code, you have the codon, and you have the anticodon. So starting with the triplet code, this is a group of 3 very specific nucleotides in a particular sequence on the DNA that are going to code for a particular amino acid. So one triplet code on the DNA encodes one amino acid that will end up in the polypeptide chain. Next, we have the codon. Similar to the triplet code, the codon is also a group of 3 specific nucleotides in a particular sequence, but instead of the DNA, the codon is on the mRNA. Each of these codons is going to be complementary to a specific triplet code on the DNA, and it will encode the same amino acid as that triplet code. So a triplet code will be transcribed into a complementary codon on the mRNA, and the codon will be translated into a specific amino acid in the polypeptide chain. This is how the sequence of nucleotides on the DNA is ending up to determine the sequence of amino acids in the polypeptide. Now, this third term, the anticodon, is a specific sequence of 3 nucleotides that is going to be complementary to the codons. These anticodons are going to be found on something called transfer RNA, or tRNA, which is something that's involved in translation. We'll talk about this in more detail when we talk about translation. For now, just remember that the anticodon on the tRNA is going to be complementary to the codon on the mRNA, and each anticodon, like the codon and triplet code, is going to be specific for the one particular amino acid. So the triplet code on the DNA, its complementary codon on the mRNA, and its complementary anticodon on the tRNA are all going to be specific for the same amino acid. Okay, so you get how we go from the sequence of nucleotides in the DNA to the sequence of amino acids in the polypeptide. But now, there's another question. How does the cell know where a particular polypeptide chain starts and where it stops? For this, there are specific codons on the mRNA called start codons and stop codons. We'll talk about these in more detail in other lessons when we discuss transcription and translation. For now, just understand that these act as indicators that will tell the cell that for a particular polypeptide, where you should start transcription and where you should stop. So it will start where there's a start codon, and when it gets to a stop codon, it will stop. Now, as we mentioned, the DNA contains genes. Each gene consists of a specific sequence of nucleotides that is going to code for a single polypeptide. So, for example, this gene here, which is represented by this red band, could code for this polypeptide. Now, our DNA contains many different genes, and each of these genes is made of many different nucleotides. A group of 3 of these consecutive nucleotides in a specific sequence are called a triplet code, and each of these triplet codes encodes a single specific amino acid. So this way, the sequence of triplet codes in the DNA translates to a sequence of amino acids in the polypeptide. Now, during the process of transcription, an mRNA molecule is made using DNA. This mRNA is going to have a complementary sequence of nucleotides to that which is on the DNA strand. So, for example, this triplet code on the DNA will be transcribed to a complementary codon on the mRNA. Both this triplet code and this codon encode the same amino acid. For example, the triplet code ATG, which is adenine, thymine, guanine, on the DNA codes for the amino acid tyrosine. Its complementary codon, which would be UAC on the mRNA, will also code for tyrosine. Remember here, adenine is pairing up with uracil on the mRNA. Since RNA doesn't have thymine, so it's going to be adenine with uracil. The thymine is pairing up with an adenine and the guanine with a cytosine. Now, this codon on the mRNA will itself have a complementary anticodon on a tRNA molecule, and that will also be a sequence of 3 nucleotides. The tRNA with the complementary anticodon will carry the specific amino acid, which in this case was tyrosine, to be attached to the polypeptide chain during the process of translation. This way, the information that was in the DNA in the form of a sequence of nucleotides is preserved and passed on from inside the nucleus to the outside where the protein is being synthesized. This is how the nucleus is controlling the activity of the cell, because the nucleus has the DNA, which has the information that encodes how to make these proteins. This process of taking this information from the DNA in the nucleus and then transcribing it into mRNA and then translating it using tRNA to form a polypeptide chain and therefore form proteins is the central dogma. This is how information from the nucleus is preserved and transferred out of the cell to be used to make proteins. Now, this information in the DNA, this genetic code, is universal, which means that all organisms that we know of have the same genetic code. All organisms that we have yet discovered have the same nitrogenous bases incorporated into their nucleotides. So the same adenine with thymine or uracil, the same cytosine with guanine. They all have these same bases, and they all pair up the same way in all organisms that we have ever found. This is why we say that the genetic code, as far as we know, seems to be universal. This fact actually comes in very handy when it comes to genetic engineering, because thanks to this, we can use one organism to make products for an entirely different organism. Having all the different organisms share the same genetic code makes doing this kind of thing a lot easier. Alright, as a quick recap, synthesizing DNA and proteins are essential processes for life to exist. The process of DNA synthesis occurs during the S phase of interphase, and the process of protein synthesis occurs during the G1 and G2 phases of interphase. Now, DNA and RNA are nucleic acids and are made up of nucleotides, while proteins are made up of amino acids. The DNA consists of many genes, each of which is made up of a specific sequence of nucleotides that encodes a particular polypeptide. The sequence of nucleotides in the gene determines the sequence of amino acids in the polypeptide. This way, the information in the DNA ultimately controls almost everything about an organism. When DNA is transcribed into mRNA, and then mRNA is translated to form a polypeptide chain, this is termed the central dogma. From this, we get the primary structure of a polypeptide, which is the sequence of amino acids. This then gives rise to the secondary structure, which is held in place by hydrogen bonds between the amino groups and the carboxyl groups of the different amino acids. This secondary structure commonly results in the formation of alpha helices and beta-pleated sheets. Then you have the formation of the tertiary structure, which involves the interaction between the different R groups of the amino acids and gives the protein a 3D shape. Finally, if there's more than one polypeptide, or if a non-protein component is added to a polypeptide, then this gives it a quaternary structure. Now, when DNA is transcribed into mRNA, the triplet codes on the DNA, each of which codes for a single specific amino acid, is transcribed to the codons on the mRNA. The respective codon on the mRNA will also code for the same amino acid as the triplet code on the DNA. Likewise, this codon will have a complementary anticodon on a tRNA, and the tRNA with the complementary anticodon will carry the specific amino acid that they were coding for to attach it to the polypeptide chain. This way, the information in the nucleus is preserved and transferred out of the nucleus to synthesize proteins, and this is why the nucleus is considered the control center of the cell. Finally, we saw how the genetic code is universal, which means that all organisms have the same bases in their nucleotides. This proves very useful for things like genetic engineering, where one organism can be used to make products for another. And that brings us to the end of this lesson. I hope you found it helpful, and I'll see you in the next one.